Thank you very much. Hello, everyone. First of all, I would like to thank HFSP not only for putting together this fantastic meeting, but also for giving us the opportunity to present our work, but also because of, what was just mentioned, it is the long-term fellowship that allowed me to develop my own independent project when I was a postdoc, which then I took to start my lab. But also the Career Development Award when they still existed, because that allowed the lab to survive basically until we cracked the grants and we got some other grants. So thank you indeed. The second thing is that even though I'm now at the University of Cambridge, most of the work I'm going to show was done here at Monash University in Melbourne when I was at the Australian Regenerative Medicine Institute. So the title didn't tell you much. So basically the idea is this classic developmental biology question of how is the control of organ size and proportions enabled in our bodies. And if you think about it, at the end of the day, it's the combination of individual cell behaviors that leads to form an organ of the right size and proportions. So the question really is, how do cells in a developing organ know, quotation marks, how big the organ is, but also how big it needs to be? And we use the vertebrate limb as a model. First of all, as Isha told you yesterday, because there is a broad range of sizes and proportions across species that we can use to our advantage. Second, because it's composed of multiple tissues that have to grow in a coordinated manner. So we have cartilage, we have bone, we have muscle, tendons, dermis, nerves, blood vessels, you name it. They all have to grow in synchrony, which is not so obvious. And also, as Louis Volper used to do, if you extend your arms like this, besides the intrinsic beauty of the limbs, one of the things that strikes you is how symmetric they are, how equal in size they are. And that's not obvious. And of course, there will be someone in the audience saying, well, actually, my limbs are not so symmetric. But yeah, okay, that's true. But in most cases, actually, the precision is between 0.2%, which is remarkable. So the two approaches in the lab is, one of them is to develop interspecies chimeras, as Isha, a postdoc fellow, told you yesterday. And the other one, which is a bit more developed, is to induce perturbation transiently and then study the recovery. So we can induce, for example, asymmetries affecting the left limb and not the right, and then we study how that recovers. So I'm going to tell you about that, and that process is called catch-up growth. So this is a normal growth trajectory of size over time for a given organ. Sometimes it happens that there can be a perturbation that deviates from that trajectory, and then in many cases, either partially or totally, there is a recovery of that trajectory. So the questions, and that's called catch-up growth. So the questions are, how is that injury, how is that defect detected, and how that triggers the recovery? But second, and equally important, is once the system is in overdrive, why does it stop? Why does it recover just the missed size without overshooting? Okay? So we use the long bones with the limbs to study this process, and long bones grow by a process called endochondral ossification. This means that a transient cartilage scaffold, here in blue, is first formed and afterwards replaced by bone. And cartilage is composed of cells called chondrocytes, which undergo subsequent differentiation states, first from round resting quiescent chondrocytes, then proliferative cells arranged in columns that push the bone or the cartilage, and then hypertrophic cells, which eventually die after laying down the extracellular matrix, the scaffold that will be replaced by bone. So bone here in red will keep growing as long as the cartilage in blue keeps providing that scaffold to be replaced. Funny thing about the cartilage progenitors or chondroprogenitors is that during fetal or neonatal stages, there is no self-renewal of those progenitors. So once they become proliferative and start forming columns, they are kind of expelled from the resting zone, and they only form short-lived columns, which are eventually pushed out of the cartilage. It's only later postnatally that they acquire self-renewal capabilities, and they remain in the resting zone while producing very long columns across the whole cartilage or growth plate, as it is called. So the question is, when and where do the long-lived chondroprogenitors arise? So were they still, or were they already in the cartilage, or were they somewhere else? And also, what happens if there is a perturbation, especially during these fetal neonatal stages when there is no cell renewal? So in order to do that, we developed this approach, which is called doxycycline control and recombinase activated gene overexpression, or DRAGON. And of course, I came up with the acronym first, and then I fit the name into it. But in short, what it means is that in order to express a gene of interest, you need two drivers. You need CRE to get rid of this TD tomato stop cassette, and you need TTA or RTTA to act on the state responsive element. So if you only have RTTA, you have expression of the TD tomato, but nothing else because of the stop. If you only have CRE, you remove the stop cassette, so it's primed for expression, but nothing happens, there is no transcription. So it's only the cells that combine, and this is the main message, it's only the cells that combine CRE and RTTA that will express the gene of interest, okay? So we can combine this with a left-specific CRE, which is expressed or acts on the precursors of the left limb only, not the right. We can combine that with a cartilage RTTA, so the early template is affected, to express the gene of interest only in the left cartilage and only TD tomato on the right as an internal control. Also, by titrating doxycycline, which is the drug that controls RTTA activity, we can do salt and pepper activation so that we have some cells in the targeted tissue expressing the gene of interest and others that don't. So we can study cell autonomous effect within the insulated cells. We can study short-range non-autonomous effect within cells of the targeted tissue. And we can study long-range communication with the surrounding tissues as well. The typical genes of interest that we express is diphtheria toxin to kill cells, or P21 to block proliferation. And as I said, because it depends on doxycycline, it's inducible and reversible, so we can control the timing. Okay, so this is a story which is under revision now. It's also on bioRxiv if you are curious. And this is the work of several heroes. So Cindy is a research assistant in the Monash lab, and she's also very good and started her own project. Then joined by Exan, a PhD student who just finished, and Ashik in Cambridge, a master's student, and also Shani and David, the bioinformaticians, because they do their magic with the data, and that was critical for this project. This all started during my postdoc, when I developed this Dragon P21 model, with which we could overexpress P21 here in blue in the left cartilage, and not so much on the right, as you can see here. This is P21 expression in the muscle as endogenous expression. But you can see here that, as I said, maybe over 60% of the chondrocytes express P21, but there are still some chondrocytes which are wild type. They only express the TD tomato, and that's because the CRE is not 100% efficient. So this kind of mosaic allows us to compare the behavior. So you can see here that P21 positive cells did not incorporate EDU, so they were not proliferating as expected, whereas the P21 negative cells were able to do it. So to our surprise, despite blocking proliferation in over 60% of the chondrocytes of the left limb, we didn't find asymmetries, which we do find if we kill cells, by the way. So it was quite interesting because obviously some compensation was going on. So long story short, what we found is that when the P21 negative cells in the left cartilage see their neighbors which cannot proliferate, they proliferate more. See, this is the left cells which are P21 negative, so these are wild-type cells. They proliferate more than equivalent cells on the right limb and more than cells in the control littermates. And the question is, what are the signals involved? How do they find out that they need to do this? And also whether the reparative cells belong to a specific population or any cell can do it. So we did single nuclei RNA-seq comparing left and right limbs of experimental animals, also with control animals. And by doing SENIC, which is a analysis of regulon of transcription factor control pathways, we noticed that the Gli1 regulon was overactivated in the experimental left limb as compared to the others. And this was interesting because Gli1 has been shown to be, or mark, the osteogenic progenitors, which are important for bone formation and repair. So we wondered whether these progenitors could also be important for the phenomenon, the compensation we were seeing. So in this case, what we did is to be able to use the CRE for lineage tracing, which I will explain in a minute. We turned to a different model in which expression of P21 only depends on the cartilage-driven RTTA, so it's expressed in all the cartilage elements. And then we use a Gli1 CRE-ER to trace the lineage. So CRE-ER is activated by tamoxifen, and that allows the CRE to act and activate a reporter. And once you do it, their modification is inherited by the whole lineage, even if tamoxifen is no longer present or even if Gli1 is no longer present. So So that allows us to trace the lineage. Okay? So by doing that, we were able to combine this lineage tracing with either normal animals or animals overexpressing P21. And we found here, this is a T tomato in situ, that there is expansion of the number of lineage trace cells. And this is quantified here at multiple stages after giving the doxycycline and the tamoxifen, with the most notable difference at P0. So are these cells involved in the recovery? In order to do that, we turn to yet another dragon allele. In this case, it's to activate diphtheria toxin in response to CRE and RTTA. So basically what we did is to combine this cartilage P21 model I explained you before with a Gli1 CRE-R, a cartilage-driven RTTA, and the DTA model. So that the Gli1 lineage here in red will be ablated, but only once they become chondrocytes, only once they become cartilage. So it's very specific targeting. This works as expected, so we detected tunnel-positive cells, so apoptotic cells, only in this triple combination as compared to P21 expression only, for example. And this is a quantification of bone length at P0, so just a few days after activating this. And you can see in this graph that only the animals that combine P21 and in which we kill the Gli1-derived chondrocytes, actually we have a defect in bone length. Okay, so what about normal growth? We have proven that these Gli1-derived chondroprogenitors are important for compensation. What about normal? Because during normal development, Gli1 is expressed within the cartilage, but also in other tissues. So we decided to label them using, again, tamoxifen-driven lineage tracing at embryonic day 13.5 or 14.5, so in the last week of gestation. And very early on, I don't know if you can see very well, but it's mostly expressed outside the cartilage and a few of them inside. But if you wait for 1 month or 2 months postnatally, actually up to 60–70% of the chondrocytes are labeled. And not only that, but if you use a multicolor reporter so that you can distinguish different clones, so cells derived from the same progenitor, you can see these long columns that I told you early in the introduction that are a feature of these long-lived chondroprogenitors. So by labeling these fetal progenitors, we were able to eventually label the long-lived progenitors. We found the origin of those. And that the question is, because Gli1 is expressed both inside and outside, whether these long-lived chondroprogenitors were already present in the fetal cartilage or were somewhere else. And I don't have time to show you all the evidence, but we concluded, or the evidence was pointing to the presence of at least part of these progenitors outside the cartilage. So we decided to turn to this marker, Pdgfra receptor alpha, as a marker which is expressed basically in most of the limb mesenchyme, but not inside the cartilage. And when you trace the lineage of those cells just in normal development, some of those cells the majority are outside, but some of those cells end up inside the cartilage. So it was the perfect tool to test, in the presence of the cartilage-driven P21, what happens with that lineage and with Gli1 expression. And long story short, what we saw is that even though there is not a big change in the Pdgfra receptor-only cells, the cells that are Pdgfra receptor lineage and positive for Gli1 expression, here with HCR, are the ones that change a lot in terms of fold change when you overexpress P21, along with the cells that are only Gli1 expressing. So at least part of the Gli1 chondrocytes are derived from the Pdgfra receptor alpha cells. Finally, to see or to reveal how that happens, we did another single-cell RNA-seq, in this case separating TD tomato positive and negative cells in control and experimental animals. And long story short, by doing pseudo-bulk analysis, we found some interesting expression changes. One of them was CCN2, or connective tissue growth factor. It is, which was downregulated in the experimental animals, as you can see here. So CCN2 is normally expressed in the resting, proliferative, and in the transition between cartilage and bone, and it was heavily downregulated, and this is quantified here, in the experimental animals. This was interesting because if we overexpress CCN2 using our dragon approach in the left cartilage, we see a reduction in proliferation, Ki67 positive cells. We see a reduction, extreme reduction in the hypertrophic zone of the left cartilage, and reduction in bone growth. So downregulation of CCN2 was supposed to do the opposite and drive the recovery that we saw. Finally, we overexpress or expose ex vivo culture bones with CCN2. we saw reduction in the number of Gli1 positive cells. So this comes to my last slide, in which we have this model where we think that when there is a normal growth output, there is a certain levels of CTGF or connective tissue growth factor in different regions of the cartilage, which keep Gli1 expression at bay in the Pdgf receptor alpha population outside the cartilage. But if there is reduced growth output due to the cartilage P21 overexpression, there is decreased expression of CTGF, as I showed you before, and that probably unleashes Gli1 expression within the Pdgf receptor alpha population, allowing them to become chondroprogenitors that then help with the recovery. Last but not least, I would like to say thank you to both labs, the Melbourne and the Cambridge lab, to all the collaborators, the funding sources, and of course, thank you for your attention.